All right, let's talk some more details about neurotransmitters and synapses. And first about the speed of electrical transmissions in nerves. So there has been selection for more rapid transmission of action potentials in axons. One way that speed can be increased is by increasing the diameter of the axon, which would make huge fat nerves. And that is not the path that has been taken in vertebrates. But in some invertebrates, we can see very large, wide axons where the single cell is quite wide. In vertebrates, axons are insulated by a myelin sheath, which is what the Schwann cells are doing in the peripheral nervous system, and oligodendrocytes in the central nervous system. And these help speed up the transmission of the action potential. In squid and cephalopods, they have the widest axons that are known, and in fact, a popular physiology lab, probably still is, to dissect out this axon from a freshly killed squid, and you can actually put electrodes in it and measure the membrane potential, measure the action potential when it happens, and it's pretty cool because they're so large. And you can see here this forcep or probe end here is about the size of an end of a toothpick, and you can see how large this is a single cell. This is a single axon here. Now in our cells and other vertebrates, there's another interesting evolutionary adaptation that has occurred, and that is that these specialized cells, the Schwann cells in the peripheral nervous system, wrap around the axons and confine the sodium channels, the voltage-gated sodium channels and potassium channels, to just these little gaps in between the Schwann cells. And this is what it looks like through an electron microscope. That's a slice through a nerve in the electron microscope. And you can see the layers of that Schwann cell wrapped around that axon. Now, the Schwann cells are living. And the little spaces in between the Schwann cells are called the nodes of Ranvier, because Ranvier is French. So, you know, French hate to pronounce the letters at the ends of words. And this is going to mean that the action potential is going to jump from gap to gap because there aren't any sodium or potassium voltage-gated channels in between, underneath that Schwann cell. So what this does is makes instead of having to go along the entire length of the axon bit by bit, instead the action potential jumps from gap to gap to gap, and that means that it goes faster. So this is the adaptation in vertebrates for the very long axons that can be present in vertebrates to have that the nerve signal move very quickly. And this is called saltatory conduction because salt, that prefix there in Latin means jump. So it jumps from node to node to node, and so that speeds up the process. So the two kinds of synapses by which neurons can connect to other cells are electrical synapses, where the action potential is going to directly be conducted from one neuron to the other through gap junctions. These are more unusual, and they're mostly in the central nervous system in the brain. Most synapses are chemical synapses, where some kind of a chemical is going to be released into the gap between the two neurons. The axon of one neuron attached to the dendrite of another neuron, and that's going to be what's going to determine whether there is a depolarization in the receiving neuron. So most of them are going to be chemical synapses. So let's talk a little bit about those. So here is the axon. The signal is coming down to the end of the axon here, and a chemical is going to be released into the synaptic cleft. The vesicles are triggered to release their neurotransmitter by another ion, calcium ions. Voltage-gated calcium ion channels open, calcium flows in. That triggers the vesicles to fuse with the outer membrane and release the neurotransmitter into the synaptic cleft. That neurotransmitter will bind to receptors on the receiving side of the synapse and trigger those ion channels to open. So ligand-gated ion channels. So instead of voltage-gated channels, so a voltage-gated channel is going to respond to the change in membrane potential. Ligand-gated channels, a ligand is something that binds to a receptor. So a ligand-gated channel means that something's going to bind to a receptor, and that's going to trigger a shape change and allow ions to now flow through by diffusion, flow down their concentration gradient from high to low. Both sodium and potassium could flow across the membrane in that case. So we call the molecule a neurotransmitter, and if enough of these synapses release enough neurotransmitter, this could trigger an action potential in the receiving neuron. An action potential in the axon is what triggers the release of that neurotransmitter, and then it diffuses into the synaptic cleft. This is a cool neuron here. So here's the axon and the ends of the axon and the dendrites over here. The receiving end in the cell body, you can see is right there. So the postsynaptic potential is going to be changed by how many of these synapses are releasing neurotransmitters. So remember that each neuron is going to have multiple connections to its dendrites, and a single signal from one synapse is probably not going to be enough to trigger an action potential. They're going to be additive. So generally, neurotransmitters that are released are going to be depolarizing in the receiving neuron. They're going to make it closer to an action potential actually occurring in the receiving neuron. Okay, let's watch this little video here. The videos for this chapter are actually pretty good.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
So coming into any one particular neuron are two kinds of signals, excitatory and inhibitory. And we already talked about both of these in relation to whether they are depolarizing or hyperpolarizing. So something that's depolarizing or a depolarization, that means it's heading towards an action potential. It's getting closer to the threshold for triggering an action potential. That's excitatory. If it's inhibitory, that means it's hyperpolarizing the membrane, making it less likely to fire an action potential, taking it further away from firing an action potential. And these signals, of which, a single neuron here was shown in this scanning electron micrograph, there are dozens of different axons terminating at this particular neuron and connecting to it. So there could be a mixture of excitatory and inhibitory signals coming in, and they're going to add up spatially in place. And they're also, if they come in at the same time, and they're also going to add up temporally, meaning if they're close together in time, they will add up and either be more excitatory or more inhibitory. But if the same two signals are further apart in time, they may not add up to an action potential. So, and we call this process summation. So any particular signal coming into a neuron from a single synapse is probably too small to trigger an action potential in that particular neuron. So it's the summation of all of the signals coming into that particular neuron at any particular moment in time. So we can have temporal summation. So this means they're close together. They're coming in, temporal means time. So they're coming in close together in time. So one depolarization is still present while a second excitatory depolarizing signal comes in from another synapse nearby. Spatial summation means that they are on the same neuron adding together. They're spatially close together. And so that means that if you have a whole bunch of excitatory signals coming in at the same time, they're going to add up to create an action potential, and more likely that that particular neuron is going to pass on an action potential to the cells that it connects to. So this summation process could also be adding up some inhibitory and some excitatory signals. So if one neuron is receiving inhibitory signals from some of its synapses and excitatory signals, and remember excitatory means depolarizing, those added together are going to determine whether that particular neuron's axon hillock is going to reach its threshold to generate an action potential. If it doesn't generate an action potential, then the cells that are downstream from that particular axon aren't going to receive any signal. So here we have two excitatory signals. E1 and E2 are supposed to be excitatory signals. You can see they're depolarizing signals. If they're far apart in time, neither one is enough to generate an action potential because they don't reach the threshold, their sub-threshold level. But if you have the same exact two signals, but now they occur closer in time, that could be enough to generate an action potential because now they're going to add together if they're close in time. And we have the same thing spatially. If we have two that are close together and occur at the same time, they're going to add up to an action potential. If they're far away from each other on different parts of the neuron, they're not going to add up to being an action potential. So the end of the signal happens almost immediately. So when neurotransmitter is released into the synapse, it has to be cleared out of that synapse very quickly to allow for another signal. And there's a couple of different ways that neurotransmitter is cleared from the synapse. One is that the molecules themselves can be destroyed so that they no longer bind to the receptor. And the other is that they can be taken up by the neuron that released them and reused. Messing with this process is the way many poisons and also medications work. For example, antidepressants, many of them work by blocking the reuptake of serotonin from synapses so that it remains longer in the synapse. And I think, in fact, the technical name for them are serotonin reuptake inhibitors. And poisons like sarin gas does the same thing. It prevents neurotransmitter from being cleared from the clefts. So you can see, you know, the dose is, the poison is in the dose. A little bit of it might be a medicine. Too much of it, not so good. So here we have a synaptic cleft with a neurotransmitter being released, and there are inactivating enzymes present so that as soon as the signal is released, the enzyme is already starting to break it down. And then the fragments are going to be taken up by one or both of the cells that are involved here. And then it has to be completely resynthesized to be used in this case. Another possibility is that it is taken up by one of the cells or both of the cells and removed from the synaptic cleft. That way, it is not destroyed, but instead it can be reused. So that's another method of clearing these molecules from the cleft. But if they are not cleared, then a new signal cannot be transmitted, or what will happen is these ligand-gated channels will remain stuck open, and that also is not good. That is not the intention of this system. We talked a little bit about one particular neurotransmitter, acetylcholine, that this is the neurotransmitter that's used by our motor neurons to signal muscle cells, and it's used in a few other places as well. There are different receptors for for the same neurotransmitter that have a different effect. So different cells can react to the same neurotransmitter differently just by having a different type of receptor that receives the signal. So instead of having a ligand-gated channel like there is in muscle cells, it could be a different kind of receptor that's going to have a different cellular reaction to the same molecule. So the two main kind of receptors for neurotransmitters are ligand-gated channels, and that's what we've already been talking about, where when the neurotransmitter binds, the channel opens and allows some kind of an ion to flow across the membrane. The other one is metabotropic, and this means that it's going to bind to the cell and the effect is going to be something else. There's going to be a G protein activated. There's going to be second messengers made. It's going to trigger some kind of a response in the cell, but it's not it's not going to be to allow ions to rush across the membrane through a channel that's going to open. It's going to be something else in the cell. That's a metabotropic receptor. There are toxins that specifically affect the acetylcholine system, like botulinum toxin. And in fact, that's you might not know that Botox is short for botulism toxin or botulinum toxin, which is the toxin that kills you if you get botulism. It's made by a bacteria. And one of the things that it does is paralyzes muscles by disrupting this acetylcholine neurotransmission, because our motor neurons use acetylcholine, including the motor neurons that innervate the muscles in your face. They also use Botox for a lot of clinical things, not just for cosmetics, like people who have horrendous muscle spasms, people who are paraplegics and quadriplegics who have muscle spasms that are causing them problems. They can use Botox injections to paralyze the muscles that are spasming and make them more comfortable. So there's a lot of uses for it. But that's one of the ways that it kills you as well, is by paralyzing your muscles. We have about 100 known neurotransmitters, and we'll talk about a few of them here. They fall mostly into 4 classes, some kind of an amino acid. There are some that are the actual amino acids that are used to make proteins, and some are modified amino acids. Biogenic amines, neuropeptides, and these are made from amino acids again, and gases like nitric oxide, which we already talked about. So here's the 4 categories. We'll talk about these in a little bit of detail, but I wanted to put the table in here for your study purposes. So amino acids are just that. And remember that although we use 20 amino acids to make proteins, there are thousands of possible amino acids, because an amino acid just means it has a carboxylic acid group and an amino group on the molecule. It doesn't have to be one of the 20 amino acids that we use to make proteins. So glutamate and glycine are amino acids that we use to make proteins, and they also function as neurotransmitters. And interestingly, glutamate is also used as a fuel source. It's been implicated in cancer metabolism as a desired fuel source by cancer cells, so it has a lot of interesting things about it. GABA is a modified amino acid, so it's not one that we use for making proteins, but it's found in the brain, it's produced in the brain, and it's used as a neurotransmitter. It's a small molecule. Biogenic amines, so again, it's an amine because it has an amino group on it somewhere. And some of these would be norepinephrine and epinephrine. Remember, these are very similar, and they differ in which parts of the body they bind to. Dopamine, which is active in the brain, and serotonin as well. Serotonin is implicated in depression and mood disorders. Dopamine is involved with voluntary muscle movements and also mood. Parkinson's disease is associated with a specific lack of dopamine in one particular area of the brain. So those are biogenic amines. And there are also the endogenous cannabinoids that bind to the cannabinoid receptors in the brain, kind of one of the more recently discovered neurotransmitters, also biogenic amines. Neuropeptides, that means that they are short chains of amino acids. So remember from biochemistry, if it's long, if it's more than 100 amino acids, generally we call it a protein. And if it's shorter than 100 amino acids, we tend to call it a peptide. So neuropeptides, that means they're less than 100 amino acids, and they are made by neurons. That's why they're called neuropeptides. And a few of those would include substance P, which is a recently discovered one, and endorphins, which have been known since the 1980s. And interestingly, endorphins were discovered because of opiates. So opiates are pain medicines that are originally, they were derived from the poppy, but we now have synthetic ones like fentanyl. But it was known since, I think, the 1940s or 50s that they were binding to receptors in the central nervous system. And so if there's a receptor for something in the central nervous system, that means your brain is making something that binds to that receptor, or something in the body is. So they didn't discover the natural substance that was binding to these opiate receptors until the 1970s or 80s. I think it was late 1970s. And these are also in the body. They are naturally occurring painkillers. And they're also involved with mood. And the same thing happened with the cannabinoid receptors, is that it was known that the substance that's found in marijuana was binding to a receptor in the brain. And if it's binding to a receptor in the brain, that means there's a natural substance that binds to that receptor as well, something the brain makes. And we call those cannabinoids, and there's several of those that have now been discovered, and that's a much more recent, that's in the last 20 years those have been discovered. So the receptor was known for a really long time, and then finally it was discovered the naturally occurring molecule that binds to that receptor, just like with opiates. Opiates and the receptors were known for decades before the natural substance, the endorphins, were discovered that bind to that receptor. We talked a little bit about nitric oxide already and its activity. This as well was not, gases were not thought to be involved in this way until the 1980s. And now we also know that, weirdly, carbon monoxide is also used in very small amounts as a signaling molecule, as a neurotransmitter as well, which is just completely weird but true. Okay, so in this chapter, we talked about the three main categories of neurons: motor neurons, sensory neurons, and interneurons. And we're going to talk a lot more about motor and sensory neurons in the next two chapters. The biggest thing to get out of this chapter is how an action potential occurs. So what membrane potential is, which molecules are involved, what do we mean by depolarization, hyperpolarization, and what exactly is happening when an action potential occurs. What are those voltage-gated channels that open when an action potential occurs? The difference between electrical and chemical synapses, excitatory and inhibitory synapses, Schwann cells in the nodes of Ranvier and how that increases the speed of action potentials, and then the different types of neurotransmitter and what are the examples of those. And what are they? So a neuropeptide, you should know a neuropeptide is a polypeptide that's made out of a chain of amino acids, and that means it's made in the ribosomes, and that's how it ends up being secreted by the cell. Okay, so that's the end of neuron, and we're going to talk a lot more about the nervous system in the next two chapters.